In the bioreactor lab, a particular strain of E. coli was cultured in a bioreactor and with inoculated flasks on a benchtop shaker. The purpose of this experiment was to compare the growth parameters in TSB media versus M9 media. Samples were taken in liquid form from each vessel and analyzed for optical density and a spectrophotometer. After taking samples for 6 hours, we were able to construct a bacterial growth curve to determine the growth parameters of the E. coli in the bioreactor and the inoculated.
The three equations on screen govern the kinetic parameters for the growth of E. coli. X denotes biomass concentration, S denotes substrate concentration, mu denotes the growth parameter where mu max is the maximum growth rate, Ks is the Monod constant, and Y sub xs is the maintenance coefficient, which is approximately 0.5. The third equation can be rearranged so that mu can be solved for using the Monod constant and the substrate concentration, as well as mu max.
Since we were dealing with E. coli, we had some extra safety precautions to follow. Gloves, goggles, and lab coats were worn at all times during the lab. All gloves had to be disposed of in a separate trash bag. We cleaned our gloves and the countertops regularly with 70% ethanol, and all samples of E. coli had to be disposed of in a separate bottle of ethanol waste. To avoid contamination, the bioreactor and all glassware were cleaned thoroughly with soap and water. To ensure that all the equipment was sterilized, the bioreactor, TSB media, and all glassware were autoclaved at 120 degrees Celsius. We made 3 liters of TSB media at a ratio of 30 grams per liter of distilled water. On the day before running the experiment, the bioreactor was assembled and flasks of media were prepared for inoculation of E. coli. On the day of the experiment, the bioreactor was running at 37 degrees Celsius and 300 RPM, while the benchtop shakers were running at 37 degrees Celsius and 200 RPM. Approximately 1 milliliter samples were taken every 30 minutes from the sampling tube of the bioreactor and put into a cuvette. The cuvette was then placed into a spectrophotometer to measure absorbance. The benchtop shaker flask had samples measured every 30 minutes under the fume hood to avoid any contamination. If the spectrophotometer readings went above 1, then the samples were diluted in half with fresh TSB media and the absorbance readings were adjusted to reflect this. Readings were taken every 6 hours until the bioreactor had to be shut off for cleaning. Once the experiment was completed, the bioreactor and all glassware were again cleaned for the next lab group.
Ideally, in bacterial growth, there are 4 phases. The first phase is the lag phase, where growth is minimal or consistent. The second phase consists of exponential growth, where the number of cells increases dramatically. Following the exponential phase is a stationary phase, where the number of cells plateaus or semi-plateaus. Finally, the death phase occurs as cells die and the concentration of cells in the bioreactor decreases. Looking at our data, there is no initial lag phase and it appears to start initially in the exponential growth period. After 250 minutes, the concentration semi-plateaus and is consistent until the bioreactor had to be shut off. Because the bioreactor had to be stopped preemptively, there is no death phase that can be seen in the plot. To determine the maximum growth parameter, a trend line was fitted to the first 6 data points on the plot. The slope of this line was the maximum growth parameter of the E. coli, which was determined to be 0.0987 inverse minutes.
The data for the flasks show that the E. coli growth was less than it was in the bioreactor. Again, only the first 6 data points were used to determine the maximum growth parameter, which was determined to be 0.0157 inverse minutes. The error bars on the graph were determined from the standard deviation error of the 4 flasks we took samples from. Using the given M9 data, we were able to determine both 1 over mu and 1 over s. Plotting these against each other gives a graph with a linear relationship. The y-intercept is equal to the reciprocal of the maximum growth parameter, and the slope is equal to the Monod constant over maximum growth parameter. Knowing this, we were able to calculate the maximum growth parameter to be 0.01287 inverse minutes, and the Monod constant to be 51.38 grams per liter. Based on our results, it is clear that the bioreactor is more effective at promoting E. coli growth than the benchtop shaker flasks, as the maximum growth parameter was over 6 times higher in the bioreactor. The TSB media also appear to be more effective than the M9 media. Our TSB data had a maximum growth parameter that was nearly 8 times higher than the maximum growth parameter of the M9 media. Our data was affected by the fact that we were unable to run the experiment until the death phase of the E. coli. Additionally, since the bioreactor was ran at a higher RPM than both the flasks and the M9 media, the resulting growth parameters might not be comparable due to an additional variable affecting results.